are like antibodies, but they're made of nucleic acids, not proteins. Similarly to antibodies, they can bind to specific molecules, so we can say that they recognize specific molecules, and this can do things like block other molecules from binding, plug up the enzyme active sites, deliver cargo to specific locations, all these sorts of things that you can do with these aptamers, kind of similarly to what you could do with an antibody, but with the aptamer, it's going to be encoded by this short sequence of these nucleotides. So this could be RNA, DNA, or XNA, which is basically an artificial type of nucleic acid. These single strands of nucleic acid fold up into unique structures, and uniquely, these structures will be what is the kind of way in which these aptamers act. This is in contrast to other forms of nucleic acid tools, things like siRNAs or microRNAs or ASOs, so antisense oligonucleotides. All of those involve short pieces of RNA or chemically modified nucleic acids, but those are actually acting by sequence-specific binding to specific locations. In the case of the aptamers, it's not the sequence that matters directly, instead it's the structure. And of course, the structure comes from the sequence, but it is the structure itself that is actually important for doing the binding, not directly the sequence in terms of base-to-base pairing. So aptamers have benefits over antibodies in various aspects, including that they're a lot easier to make. It's easier to make lots of copies of nucleic acids than it is to make proteins. And so with proteins, you're typically dealing with cell expression, and you have to worry about proper folding. I mean, you still have to worry about proper folding with aptamer, but you're dealing with a short thing as opposed to a really long thing. With protein, there's all these complications, and it can be really expensive. You can't give it in like a sort of pill form, because then the body's going to digest it. And so various things like this that make antibodies a lot harder to deliver, keep stable, this sort of thing, than these aptamers. Now, aptamers, you still have to worry about stability, but here you have to worry about stability by things like nucleases, which are going to be enzymes that degrade nucleic acids. You also have to worry about immunogenicity, so basically the body recognizing that there's 4 nucleic acids and being like, that's not right, let's go and attack it. And these problems can be gotten around if you want to use these aptamers therapeutically by doing things like chemically modifying them, adding changes to them that don't mess with the structure, but allow them to kind of be shielded from the immune system, be more easy to deliver, be protected from nucleases, that sort of thing. And these can include modifications to both the backbone as well as the individual bases. There can also be modifications to kind of keep that structure more stable. When you're dealing with other types of nucleic acid tools, so things like your siRNAs, your microRNAs, or your ASOs, here too you're often going to have chemical modifications for the same sorts of reasons. Let's discuss really quickly some of the differences between these different types of nucleic acid therapies. So I'm going to talk mainly about these small therapies, and so we also have things like gene therapy. We're delivering whole messenger RNAs for making proteins. We can talk about doing gene editing and things. That's not what I want to talk about here. Instead, I'm going to talk about the shorter things. So we have microRNAs, siRNAs, which are kind of clumped together. We have ASOs, so antisense oligonucleotides, and we have these aptamers. As I mentioned before, the microRNAs and siRNAs, the miRNAs, we call those the microRNAs, and the siRNAs, as well as the antisense oligonucleotides, those are acting based on the sequence specificity, whereas the aptamers are acting based on their shape. With the microRNAs and the siRNAs, they act through the RNA interference or RNAi pathway. So they get bound by a protein called Argonaute, and Argonaute uses them as guides to find complementary sequences of messenger RNAs, and then Argonaute binds to those messenger RNAs through that small RNA, so that microRNA or the siRNA, and then upon binding, it's going to shut down production of protein from that messenger RNA. They can be delivered as double-stranded RNA, siRNA duplexes. They can be delivered as shRNA, so short hairpin RNAs, which can actually get like kind of encoded by the cells if you want to have a longer-term knockdown, this sort of thing. But those are acting by binding to Argonaute, and then Argonaute takes them to bind them specifically to specific locations on messenger RNA and shut down production of protein. With an ASO, there's no AGO involved, but the ASO is basically going to go and it's going to bind to a specific sequence on the messenger RNA or the pre-mRNA. These ASOs are going to be more DNA-like, but again, they're going to be chemically modified nucleic acids. And what they're going to do is they're going to bind to either messenger RNA, so mRNA, or pre-messenger RNA, pre-mRNA. If designed to bind to the mRNA, they'll bind to the mRNA, but they won't need AGO in order to do so, so it will just bind directly. Upon binding, the cells can be like, oh, there's a DNA-RNA duplex, that's not right, and so then RNase H is an enzyme that will actually cut the RNA in that case and prevent protein from being made. They can also directly just block translation through the messenger RNA. They can also block other things from binding depending on where you design them to bind. If you design them to bind on the pre-mRNA, you can have them binding at splicing junctions and direct splicing in specific ways, and this can do things like correct splicing deficits. So those are the ASOs, and then we have the microRNAs and siRNAs. And again, all of those are binding based on sequence-to-sequence pairing. And the nucleic acids that they're binding to are inside the cells, and so these have to get inside the cells as well, and there are complications with that. Now, aptamers are different. They don't depend on the sequence directly. The sequence determines how they fold, but it's that fold that then goes and makes them be active and allows them to bind to specific things. So they're not binding through that base-to-base pairing, they're binding based on shape-to-shape complementarity, not base-to-base complementarity. And so they can act on targets other than nucleic acids. They can bind to proteins, they can bind to small molecules, they can bind to all sorts of things. And this allows them to have a greater range of flexibility and be able to act outside the cell, do things like bind to extracellular proteins. So say surface receptors on cells, which are then going to allow these aptamers, even in some cases, to target direct specific kind of like payloads, so specific things to specific cell types. And so these aptamers can actually be conjugated to or basically chemically attached to various molecules in order to deliver those various molecules to different places. And so there's a lot of cool things that you can do with these aptamers. In addition to be used therapeutically for things like blocking the binding of other things to proteins, similarly to what we could do with things like monoclonal antibodies. You can also use them in the lab as things like biosensors. You can have ones that change shape upon binding to specific things. So there's all sorts of different things that you can do with these aptamers. And it's kind of a growing technology that was kind of forgotten about for a while and is now kind of coming back into vogue. And one of the reasons why it's kind of coming back into vogue is because scientists are increasingly getting better at making high throughput, so basically really fast versions of allowing you to develop these aptamers that will target different things. The main way that they do this was the method called SELEX. SELEX is a form of in vitro evolution. And so basically in a lab evolution where you are trying to make better and better versions of something until you have something that's really optimal for use. So some types of directed evolution you might see in a lab are things like phage display and various things like that. But with the SELEX, here you're dealing with nucleic acids, not with proteins or whole cells or things. So with nucleic acids, things are simpler because you are just dealing with these short strands of nucleic acids that we have methods in order to amplify and make lots of copies of, that is. And so in SELEX, the basic idea is that you have this pool, this library of aptamers, so a bunch of these different aptamers. And then you take them through these cycles of testing if those aptamers will bind to a molecule of interest, isolating the ones that do bind, mutating them, so slightly changing their sequence, and then seeing if they bind better, and then taking the ones that bind the best and keep doing this over and over and over and over and over until you get the optimal binders. And then once you get those optimal binders, then you can do things like optimize them for features for delivery, lack of immunogenicity, stability, and things, and then make sure that they still bind the way that you thought they would before. Similarly to antibodies, aptamers typically have unique parts and variable parts. With an antibody, we kind of have that constant region, which is kind of like the bottom of the Y, if you think about your classical like IgG antibody, and then the variable regions, the parts at the top that differ and bind to different things. I like to think of that constant region as a kind of adapter, which allows different variable regions, which recognize different things, to be placed on the same kind of adapter that then allows cells to deal with all of these different antibodies in the same sorts of ways, but all those different antibodies would recognize different things. In the case of the aptamers, the unique parts are kind of in the center, not on the ends, and the constant regions are on the ends. And so if you think about an aptamer, you have the short strand nucleic acids, and then at the ends you have these adapter regions, which are going to be constant regions. And so those are going to be the same in all of these aptamers. And the inside part is going to be the part that differs, and that differing is going to allow it to fold in different ways. But having these constant regions on the end is going to allow them to serve as sort of adapters, which are going to allow you to easily make lots and lots of copies of these using PCR, so polymerase chain reaction. And so basically, if you want to make a lot of copies of the DNA version of it, what you can do is you can use PCR and use primers, so these short oligonucleotides that direct the copying machinery that match those adapter parts on the end, and those adapter parts are going to be the same for all of your aptamers, and so you can easily make lots and lots of copies of your entire library and lots of copies of the ones that you select that bound. And then there are various techniques that you can use, like error-prone PCR and chemical mutagenesis, in order to make changes to the sequences in those libraries, and then do this again and again and again. Now, if you want RNA, then you're going to have to do things like actually do in vitro transcription instead of just PCR, but you can still use PCR to make the templates. So there are various things like that, but having these adapter regions on the ends is going to be helpful. And it's also going to be helpful because then it's going to allow you to more easily conjugate or attach these aptamers to various things if that's what you want to do. But the basic idea is that then you're changing the inside part of it, seeing if it binds better. And by having these adapters, you're able to easily isolate the things that you want and make lots of copies of them. So aptamers, again, are different from other forms of nucleic acid therapies and tools in that they bind based on their shape, not based directly on their sequence and base-to-base pairing. As such, they can act on a variety of different molecules, things like proteins even, as compared to the other nucleic acids, microRNAs, siRNAs, ASOs, which are restricted to what they can bind to, and because they can only bind to those nucleic acids. And those nucleic acids are going to be found inside of cells, and so they can't act on like extracellular targets, whereas these aptamers can. These aptamers have benefits over antibodies in some manners in that they're smaller, they're easier to manufacture, they're often more stable when we do these chemical modifications, they're easier to deliver, and they can be conjugated directly to a bunch of different molecules. You can easily evolve them, and these sorts of things are going to allow these aptamers to have a variety of benefits over other forms of tools in particular cases. And so there are a couple of examples of aptamers that are actually used therapeutically. The first ones have been for like eye diseases, and it's something that you want to keep your eye on, especially as technology improves for these high throughput, or basically able to test a lot at a time, methods like improved versions of SELEX that are going to allow scientists to more easily make aptamers that will bind to specific targets of interest. So keep your eye out on the aptamer field and hope that this was helpful.